teaching currently HMB 310, and I think one of my students is actually here, but he's muted right now. So good to see that he's here. And in the winter, I'll be teaching at HMB 200 and also HMB 312. So those are second and third year biology and the neuroscience courses.
Great. Do you have the titles of those courses for folks who may not be in human biology?
Yes, but it might be a little wrong. I know that for HMB 310, it is called Laboratory in Neuroscience.
Right.
And that HMB 200 or like 200 is Introduction to Neuroscience. And then HMB 312, like 312, is Laboratory in Health and Disease.
Health and disease, which is
Yep.
so closely related to the project you're going to talk about today, but
For
it's
sure, yeah.
wonderful. Yeah, it's wonderful to hear that you also have such a developing background in neuroscience as well. Alex, how about you? Are you teaching this semester or next semester? What are you teaching?
Yeah. Hi. Yeah. So first of all, thanks for having us. Yeah. So I'm actually teaching HMB 300 with Professor Ju, who's here. So that's neurobiology of behavior.
Fantastic. Yeah. Anything next semester, Alex?
No, not yet.
Okay, great. So now I want to hear a little bit about your own doctoral work. Alex, we'll begin with you and then we'll go to Jonathon. What university are you studying at? Are you with us or are you somewhere else? What's the name of your program?
Yeah, so I'm here at U of T. I'm with the Department of Biomedical Engineering. So that's part of the Faculty of Applied Science and Engineering. And my degree is called a Master's of Applied Science. So, yeah.
Fantastic. And how did you decide that that was the right program for you?
Yeah, so it took a lot of experiences kind of added up, but I was really interested in epilepsy research throughout my whole undergrad. So I had a couple opportunities throughout, I think mostly third and fourth year, which kind of exposed me to the research environment, types of epilepsy research, and then I actually took a year off after my fourth year where I was able to like really think about what program I wanted, I guess methods I was really interested in. And so I found my supervisor first before the program, and then he had suggested like to apply to biomedical engineering. And yeah, so that's, I guess, the path I followed.
So interesting. So you actually took some time to do some research to figure out what would be the right program, what might be the right even methodology. How did you go about doing that research? If some of our listeners today want to do that as well, how do they even begin? You took a whole year off, so you clearly had a particular journey you took.
Yeah, that's a good question. So I was more interested in, I think, finding a supervisor, because I think with thesis research, it doesn't matter too much about the program. Like there's obviously minor differences, but I think more important is the supervisor and like the lab that you're working in, because that's where you spend most of your time. So I was part of like the epilepsy researchers in Toronto have a little community called the EPLink community. So I was kind of already familiar with some of the PIs in that little group. And then, I kind of looked into the other PIs, read into their research. And also I talked to the grad students in the lab like that I currently am in. So pieced together all those things. And then I finally like met with the PI and talked to him, and he gave me some like words of advice to which program to apply to. And we kind of had an informal chat. So yeah, that's how I researched my program.
Fantastic. I'm going to talk to Jonathon for a while about the same thing. When I come back to you, I want to hear a little more about how you found those opportunities to do research as an undergrad student and why epilepsy. So you can think about that. Right. Jonathon, where are you studying? Are you with us? Are you somewhere else? And what program are you doing your doctoral research in?
Sure. So I'm also at U of T, obviously St. George campus. And then so I'm doing my program at the Institute of Medical Science, or I'll call IMS for short, and it is part of the Faculty of Medicine.
Okay. And how did you decide to do spinal cord injury research? How did you find your way to your program?
Oh boy, this is a long story. I'll save some parts for part two as well.
Perfect.
So in my research, especially in spinal cord injury, well, I was originally looking at the doing research in neuro and immunology because those are like my two favorite topics, especially as a researcher. And spinal cord injury was also something that is really important to me because I love to be like I play a lot of sports, therefore motor behavior is so important. And the cord is also a very important aspect in like helping us just like do a lot of the behaviors and the motions, right?
Right.
And so therefore, so using those two as like a framework, I'll say, is that so then I was looking at the different bosses or the what I'll say as the PIs or the principal investigators. And then when I was looking in that part, I was also looking for the PIs who do the MD PhDs or the clinician scientists, because that's also what I want to aim for in the future as well. So considering all those different aspects to it, so therefore that's why I ended up in my current lab now. Like it's a mixture of everything, like the environment, the project. Like do you want like a large or a tiny lab? Do you want your PI to always be with you or give you more autonomy? Like all these different soft traits, that aside from the grades and the marks and everything else, like this one big package. That's why I'm here today.
Fantastic. So when we get to part two, I want to hear a little bit more about how you made those decisions. You gave
Sure.
us such a great list of what to think about, but what played into your decision making. But back to Alex now. Alex, you talked a little bit about opportunities you had as an undergrad. Can you talk a little bit about how you found those opportunities and why in the end epilepsy research?
Yeah, so I was really fortunate to have, I guess, a couple, I think I had about 4 in total throughout my undergrad, which is like really, I feel lucky to have. So I started my journey through the work study program on Career Learning Network, and I had a position in a psychology lab doing EEG research. So I was running participants, which was really cool because I was interested in the brain, neuroscience, like that was my program. So I wanted to see what research was like in that field. And then I had applied for a summer internship through biomedical engineering. They have a summer student program. And I, again, with that program, I had found a researcher I was interested in. And then I saw that like he was part of the summer program. So I kind of went from there. And I first I emailed him and was just illustrated like why I'm interested in his research, why I like epilepsy, what skills I already have from my work study, and then kind of what I'm interested in learning. So I had the opportunity to work through the summer there. Then I think, oh, I also, so funny that you're a TA in HMB 310 because my other research opportunity was from a TA in HMB 310. He said he worked with this like epilepsy researcher, and like immediately I was like, okay, like I need to meet this guy. So he suggested I go to their journal club, and then I just talked to the PI, and he got me like a volunteer position. I ended up doing a research course with him the following year. So yeah, and then my last opportunity, I guess, was work study during my 4th year that turned into a full-time job when I graduated. So I could still be involved in research after I graduated, but not like rush into a master's because I didn't feel ready. Yeah, so that's kind of like, I know it's kind of a lot of things. I don't think most undergrads will have like a lot like all those opportunities, but it's definitely worthwhile to check out like CLN and that kind of stuff. Yeah.
What I love so much about your story about your journey is that it started off with a work study position, and work study positions, there are many, 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 and they are open to undergrads, and it's one of those resources that is very accessible, and it's so interesting to hear how that opened up a whole lot of other opportunities. The other thing I thought that was so interesting about your account was you talked to your TA, and here you are, you know, talking to a bigger group, but I think, you know, often undergrad students don't think about using their TAs who are, you know, just a little bit ahead of them in the academic journey as an important resource. So I think that's both of those things were terrific. Okay, Jonathon, back to you. Continue telling us a little bit about your journey, how you made those big decisions that you made to end up where you are.
Sure. So, I guess my journey began in second year when I was trying to look for research opportunities as well. So back then, because I think the biggest challenge is that when you get the experiences, like they always say, we prefer people who have experience, but then if you don't have experience, then how can you get in the first place, right? So that's like the ongoing loop of things. And so literally when I was applying for my first research assistant job in second year of university, I emailed 60 profs, like saying like, hi, I'm Jonathon. I'm interested in research. Can I join your lab for the summer to do the work, right? And then I kept getting the like no, 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 like all the way through. And then finally someone said yes, but only if I could win the NSERC, which is like a competitive award as well. So fortunately, I was able to win the NSERC, so therefore I was able to get my foot into the door and therefore start my research opportunities. But I guess it's just to say that like it's hard to get your first acceptance or like the first yes, but once you get there, it's a lot easier. So actually, because my ongoing goal was to do the research in biology, like in the neuro and the immunology. But my first experience was actually in the environmental sciences, like which is a bit different. But that does not mean it was bad because that was where I was able to learn how to read papers, like learn how to a lot of the hands-on skills, like learn how it is to be as a research student, and then be able to get these translational skills, which I'm then able to then relate to my on my CV and the resume to my future bosses, like saying like, even though I have research in the chemistry that might not be directly associated with your research, but here is what I can do for you to help your team. And so that was how I was able to to jump from like A to B to C. And then in between, I was also doing some work study as well because students are sometimes quite poor, and then so the extra money, it does help. And then for my fourth year, I was doing a undergraduate thesis so in immunology because my professor back then was actually quite new, and so because her lab was so new, she really needed someone to like really to do everything.
Yeah.
So while it was hard to like get her lab started, but in that position I learned so much, even though it was very hard. So that was like, it's like literally trial by fire. So it was a tough learning experience, but that taught me a lot. So with that immunology experience, I was then able to transition into my current lab where I do both neuro and the immunology. But as Alex pointed out before, there are also various research programs out there that, especially during the summer, that will allow you to do research that's paid at the PI's lab for over like June to August. Obviously, because of COVID, those programs were canceled, unfortunately. But that will be a great resource because not only are you able to like work full time in the lab, if you are able, if you like what you are doing, and you like the lab as well, that is a very easy way to say like, hi, I was a summer student before. I like what your research is. Are you opening to have me as a master's student? Like that's a very easy transition from an undergraduate towards a research based master or the PhD.
Fantastic. What I'm hearing from your story, Alex, is that you had a lot of confidence. And even though you went through some difficult experiences, for example, helping your prof get her lab started, and even though, you know, it was a bit of a leap for you, you had the confidence to take the risk. You had the confidence to take the risk and to learn something new. And you didn't worry too much if not everything went well. Everything is a learning experience, and even though it was hard, you knew that you would learn a lot. And I think that's a really important thing. I think often we wait until we have the experience and therefore the confidence to say I have something to offer. And in your story, what I heard is you showed whatever you had to offer and were willing to go into areas where you weren't, you know, already prepared, because how could you be until you had the experience, and move with the experience. Just before we go back to Alex, and now we're going to get specific into your research projects. You said that you emailed 60 professors, and you didn't get discouraged by the no's. You finally got a yes, but the yes came with a particular request. You had to win the NSERC. Just in case folks don't know what an NSERC is, can you tell us what that was and how did you get prepared to win that NSERC?
Okay, so NSERC, so for the, I think NSERC stands for Natural Sciences Engineering Research Council of Canada. I might be wrong there, or something along those lines. And then it's called the USRA, or the USRA Undergraduate Student Research Award. So that is a funded opportunity where the money goes to the prop's lab, and therefore the prop is able to pay you through this award. And to be able to to be able to apply for this award, it is based on both your merits, so like your GPA, your stats, and all that. But also it is how you're able to write your research proposal in a way that is understood by both the lay people, well, not lay, but like the regular public, and the scientists who are able to judge the merit of your award. And I remember that when I first wrote my research proposal, it was really bad because I was in my first year and my prof was like, what is this? Like, this is not that good. So I was like, another no, but I learned a lot because as a student, you typically just think science is just like X, Y, and Z, right? But there's so many ways that science can be perceived. And to be able to convince someone that not only are you a strong candidate, but your project is worth funding, like that's a big challenge. So that was like the initial, like the barriers I had to overcome. And also some luck as well, because I think that year, I think one person who got the award actually dropped out. So I was the next on the list. So like hard work, yes, but also some luck as well. Because like I always, the quote saying that the harder you work, the luckier you get. That's something that I live by as well, because you can work as hard as you want, but if you get unlucky, then you won't get anywhere, right? So it's a mixture of both.
It's preparation meets opportunity,
For
and
sure,
making lots of opportunities for yourself and being able to accept feedback with grace and learn from it. These are all really important things. Okay, Alex, we're back to you. We're going to get specific now. And can you tell us the title of your master's project? What's it about? If you can do it in that accessible way that Jonathon talked about. And can you also then tell us about an important moment in your research journey so far?
Sure. So I guess the title of my project is Characterization of Seizures in Organoids and Testing Medical Cannabis Compounds to treat the seizures. to, I guess, hopefully end up with some treatments in epilepsy. That's a really long title. But pretty much what my project entails is I have a seizure model. So if, for those of you who don't know, epilepsy is a neurological disorder characterized by recurrent seizures. And so about a third of patients don't respond to drugs. So it's really detrimental to their lives and quality of life, all that. So we still need to research new drugs. And so I have, as part of my project, a seizure model, which is organoid tissue, which sounds like pretty science fiction, but it's human stem cells, and they're grown into brain tissue, brain like cells. It doesn't form like a full brain. They're like millimeters big, but they're using human stem cells as opposed to like any kind of animal tissue. So it has that advantage of being more similar to human DNA, right? So I have that model, and I'll induce seizures into it so it looks like it has epilepsy. And then after that, I add cannabis compounds. So some commonly known ones are THC and CBD, and they're used to, like CBD especially is used to treat epilepsy right now. So we're testing those in combination with other types of anti-seizure drugs, other cannabinoids, which ratios, you know, interact well, which is the most effective at stopping the seizures. And then the whole overarching goal of this project is to provide hopefully some future treatment options for people with epilepsy. Yeah, so that's my project. And then what was the second part of the question?
I'll just say you did a brilliant job at making that accessible for folks who don't have that. That was great. And it's great to hear you speak so eloquently about your project. Can you talk about an important moment in your research journey so far?
Yeah, a really important moment, I was thinking about this, was when I did my experiments with organoids all by myself for the first time. And I'm pretty sure every single step went wrong. But like the first thing is the organoids, they have to be stored like at a certain temperature, certain pressure. And like the first thing I do in the morning, you know, is I go to get the organoids. And then I walk in and there's an alarm blaring in the room, and the like whatever the incubator was like broken, and I'm like, great, wonderful start to my day. Obviously the first time I'm doing this by myself is I'm gonna encounter problems like that, but kind of going through all the steps on my own, because I had only watched the other student do it, and I had never like gone through the actions, but you know, I kept like making mistakes, or like my hand pressure was too much, or I poured something too high. But that was really important because now I like know what I need to do better, how to learn, how to improve my technique. And just, it was a whole learning experience for me. If everything had gone perfectly, then I feel like I wouldn't be paying as much attention, but now I'm like, okay, this went wrong. So what am I going to fix for next time? Or like, how am I going to practice? So yeah, that was a really important day for me. Yeah.
So much in that account, you know, Alex, first of all, you know, it's easy to freak out if you come into the lab and face everything yet you were facing, but you didn't. You were calm, and even now you talk about it wasn't a great day. And then again, this is a theme that Jonathon started us off on, is that mistakes are going to happen, and actually that's how you really learn. Maybe you wouldn't be perfecting your technique as much if there hadn't been a mistake. So not letting mistakes or setbacks, you know, devastate you, paralyze you, but embrace for whatever that moment is there to teach you. Very inspiring. So great. Jonathon, we're back to you now, and we're getting specific into your project. What's the title, and what's it about? And when we're done with that, an important moment in your journey.
Sure. So that's a three-part question. So I'll answer them like one by one. So
Okay.
the title of my project is Human Immunoglobulin G, or like HIGG, to treat spinal cord injury.
Okay.
And so my project is involving in the basic sciences. So I use animal research, so the adult female rats. And then so I use that model as a way to be able to induce an injury onto their spine so that they are injured in a cord. And then I'm looking at the immune response after an injury towards their spine. And this immune response is very important because after the injury is occurred, there's a two-part process. So there's the initial crush, like the cord is damaged. And then because the cord is damaged, the cells are dying. People like not people, cells are going crazy in there. And then the immune response will indicate that something is going wrong. And then so all the immune cells will then be coming in as like the first aid responders. It's like see what is going wrong, like what can they do. And then so these immune cells, they do two main aspects that are both positive and the negative. The positive is that because they are the first aid cells, they're able to remove away the dying tissue to be able to make the injury clean and be able to secrete factors to be able to enhance the repair and growth. And like it's like able to put in a band-aid and some ointment to make you feel better, like after an injury on your own. But the thing is that if the immune cells are in there for too long, they can also cause damage. It's like when you put on bandage for too long, you put on the wrong ointment, like it will also cause damage as well, like even additional damage as well. And so therefore there is a very fine balance to be able to only let in the correct immune cells to enter into the core, so like the right ointment, and to not have too much ointment because of the effects. Mm-hmm. So right now, in the clinic, if we are to treat a patient with the with the injury in their spine, we administer a drug called the methylprednisolone, which is a global immunosuppressant. But because, as previously said, the immune cells are both good and bad, right? Mm-hmm. So ideally, you want to be able to only shut down the negative, but allow the positive to grow. But in this case, since it is a global immunosuppressive drug, it will shut down both attributes. So therefore, it's not as efficient as it can be. So therefore, my research is using the molecule called HIGG to be able to re-attain the balance of the immune cells. So we only have the proper amount and like the proper ointment putting on at the proper amount of time. So it's like Goldilocks. Like the porridge is not too cold, not too hot, like just at the right amount. So that's how I envision, like, just, and then to do that, I look what is the best dose to administer the drug, the best time point, and also how the drug works. So that's like the three aims of my PhD thesis. So what was the question 3?
Moment in your journey so far?
Oh, 7 years in grad school is a long time. To choose one moment
of this particular project, of the PhD project.
I think it would be attending my first conference.
Tell us about that.
So I was attending my first conference. It was, I think, in Kentucky, like in the States. It was my first time presenting my poster. I was in a suit, like in a pretty, like people were around with cheese and wine. Like it was pretty formal. But I had to like present my poster to a lot of the judges, people who were typically not in the field. And so some questions they were asking was like really from left field. And I was like, holy cow, like this is pretty impressive. But the fact that you're talking to people are actually interested in your work, and questions are both easy and hard. Like that was where I was able to like meet a lot of people in other fields. So therefore, the theme of not working in silo, but more of the collaboration. So therefore, you're able to meet new people from all over the world, get their feedback, like you help them and they help you. So overall, we're able to like to do better science overall. And like so after my first conference, that was like the point is like I made it. That this is how it's like to be a scientist, something that I love, and that's like what I want my career to be a part of as well.
It sounds like it was an identity, a professional identity defining moment, right?
Yeah, it was.
That's great. Alex, did you have a chance to do any conferences yet? Do you want to talk a little bit about your experience with conferences?
Um, I haven't yet, just because I've only started in September, so I don't think I have enough data yet.
Right.
But in my previous research experiences, I did do conferences, and yeah, they're a really like fun experience because, like Jonathon was saying, you get to talk to people who are, kind of outside of the field and kind of see a different perspective. And then people actually are interested in your research, and you're like, oh my God, I'm actually doing something worth something. Like sometimes as a scientist, you're like kind of in the lab, like, every day working really hard. And then when you go to a conference, it's like, oh, people actually care about, this type of work. So it's really, really fun experience and good to also get ideas, like meet new people for, research that's in your field. You'll definitely encounter maybe projects similar or supervisors doing similar work. So it's great for that too. Yeah.
Yeah, what I'm hearing from both of you is how validating it is after spending so much time alone in a lab to be able to share your results with people and see where your work fits in to not only just one community, the community that you're involved in, but other communities as well. And that's something to look forward to. I'm going to ask you both one more question, and then I'm going to warn the folks who are listening in. We are going to then have a little bit of time for questions or takeaways. Takeaways is something I use in my own class. At the end of every class, people go around and they talk about something they're taking away from that class. And so I'd like you to think about something that you're taking away from what you've heard so far, or if you have any questions based on what you heard so far. We're going to go to that right after this last set of questions. And I wanted to ask you both, who do you hope will listen to your results about your research? Who do you hope will read about your research? And why is it important for you to reach that audience? So we'll go to Jonathon first. Who's the audience for your research, Jonathon? Who do you hope will read about your stuff? And who do you hope will engage with it? And why would that be important?
I I want to say everyone, but that's like a very generic and vague answer.
Yeah.
But ideally, I want to be able to target the public because the public is where a lot of the research money comes from. Like just to be able to like explain to them like why my research is important. But also right now, especially the doctors out there, because right now like a part that I didn't mention is that after an injury towards the cord, there's also the a peripheral immune suppression, which is like when your body just gets a lot weaker. Especially can lead to the infections in the skin, the wound, and also along the um, respiratory tract, especially with COVID going on. So it's like, especially like this, it's just that like, because sometimes doctors, they always think that one disease can only like influence like one area. It's actually because it's just like, it's an entire body, right? So
Right.
one disease can actually influence a lot of things. So therefore, it's just to be able to communicate like that aspect and like how through my research, like by being able to balance the immune response, be able to establish homeostasis and not just only in the cord, but also in the entire body as well.
I love the fact that you want your research to reach the public, and you're absolutely right. All of our research is funded by public money, and we often don't name those people in terms of our audiences. And of course, the way you've explained it, it's clear why your research will be important for doctors as well. Alex, how about audience for your work? Who do you hope will learn about your research and why?
Yeah, so also like Jonathon, I wish I could say everyone,
Yeah.
but definitely like award committees are a good audience for, the product of your research. I also think what's really valuable is, especially for like medical research, the patient population that you're investigating, you really want them to be able to digest your research or make it accessible, or it should be accessible to them. So one thing I do a lot is outreach in the epilepsy community. Like there's a number of charities and like non-for-profit and events. And so there's like an annual event that it's all audiences. It's scientists, it's patients, it's funders, it's, the charity, like all these different types of people who play a role in the research. So I think that's important. Like going to those types of events will be important to broaden your reach. And also, I guess, other students who are interested in this type of research, it's good to, if they're, don't know what they want to do, talking to a grad student about their research is a good way to, I guess, get inspired. Yeah.
Fantastic. Okay, now it's time for us to open it up. If you would like to ask a question or give a takeaway, just go ahead and say, I have a question, and we'll let you go ahead. If you're more comfortable putting it in the chat, that's good too. And I have one question in the chat. We're going to, we have a few. So we'll go through the chat, and if you want to jump in, let me know. Just say I have a question once we get through the chat. Okay, and Colleen says we have at least 6 human biology students here. She wants to hear your questions, and she has named you, so the challenge is out there, and some of you have begun to take it up. The first takeaway question, it seems work study is a good way to earn experience. It seems most of them have intimidating requirements. Are skills learned in undergrad going to be enough? So can you talk a little bit about work study? Was it intimidating? And the skills that you learned through work study, were they enough to take you to the next step? Jonathon first, and then Alex.
So I believe that the undergraduate experience is enough because it will provide the right foundation to get the role, but it is how you're able to communicate what you've learned from previous experiences and apply what you've learned to this new lab or this new role that will really like get you through the door. And it is the communication aspect that is the key part, because you can have the best GPA in the world, but if you cannot explain how you're able to help the lab or the role and like how you're fit in that job, then your application might not be viewed as positively as someone else who might have a lower GPA, but they're able to communicate a lot better in that sense.
Thank you so much. Alex, was it intimidating to try and get a work study position? Did what you learn, was it enough to take you to the next step?
Yeah, I agree with Jonathon, what he said. It's about like how you talk about your skill set. So for example, I used all the skills I learned in HMB 310, the lab. I would, write a whole paragraph about that. And you don't necessarily have to be explicit in that, I was in the lab like once a week for 5 times. You can just say, I learned this skill, I learned cell culturing, and that's what they're going to look at. Also, I took a stats course in R, and one of my research opportunities actually mentioned, like, we took you on because we saw that you had R experience. So it's really about like how you sell yourself and how you talk about your experiences. Also, the work study, like, they should be accepting students who are who don't have that much experience. So try and keep an eye out for the ones who are like accepting 1st and 2nd year, but it's definitely enough to kind of get your foot in the door.
Wonderful. Next question. How feasible is it when you have a full course load to undertake a work study question without compromising your mental health or grades? It's always a balance, right? And it's so clear that work study was a big key to get you to the next place, but you had a full course load, and you need to make sure that your mental health isn't compromised. Do you want to each of you talk a little bit about that? Maybe we'll go to Alex first. Did you feel the balancing act was difficult? Do you have any strategies for managing school and work study and not compromising mental health?
Yeah, that's a really good question. And I think it took me a little bit of time to get it right, a lot of trial and error. But I like to tell people, like, with, who are doing a lot of things, there's always this like optimal amount of busyness and like optimal amount of stress. So I find, I was doing my courses and I had this work study and maybe some extracurriculars, but because I had those extra like out of academic opportunities, I was motivated to get my academic work done. So if, let's say, I have to do my work study, go into the lab on Friday, and I have something due on Friday, I absolutely get it done Thursday night. So, managing your time, Google Calendar is like every single thing in my life is on Google Calendar, so I can actually see, like, do I actually have time to do this? So time management, keeping yourself optimally busy, which is a trial and error, figuring it out process for unique for everyone. Yeah.
Great. Jonathon, I'm going to give you the next question. And the next question has to do with what you were talking about when you said you were had to apply to NSERC. You said that you had to prove to them that you were a strong candidate. What is your advice on how to present yourself as a strong candidate worth funding or joining a lab?
Sure. So I think it's also the same question for Anita. I'm not sure if I'm saying her name right. Aneta, I'm sorry about that. That wrong. I'm saying your name. But one way that you're able to, I think, highlight yourself that you're worthy of being funded by the lab is that when you write your emails to the PI, make sure that like their ongoing projects. Maybe even read a few papers, because usually in the papers, like, they'll say areas for future research, and like there's like X, Y, and Z. And if you're able to highlight, like, this is like the next thing that you want to do, and here is my skill set that I can be able to help you to do that, like, that indicates a few things. It indicates that you've taken the initiative to understand, like, really what the PI actually does. Second, you took the time to actually read their publication, and the research publication is like almost like gold. And the third part is that it really gives you a good framework to really apply your skills in the way that will really help them most. So I would do that.
So reading the papers by the profs for whom you are applying. And that does connect to Annette's question about needing to do a literature research for your project. That is research, and you ended up reading some important papers. I'm going to go to you, Alex, for the second part of Annette's question. What are the research skills, if any, you wish that you had learned while you were undergrad? Was there anything that you felt you were missing that could have been added to your undergrad experience? Or did you find, given the variety of things that you've done, you were able to develop what you needed?
Yeah, I think, like, I really enjoyed my experiences through undergrad, but I think it was attributable to the fact that I was constantly like going out and seeking opportunity. And I think that, we live in Toronto. You're at, like, I think it's #1 university in the world now, or in Canada now. There's such a richness of opportunity, but like, it's not going to just be on your plate. You really need to go out and search for it. So luckily I kind of developed that seeking attitude early on, but I would say throughout undergrad, a lot of things that will help you is if you're going, searching on CLN every day, like keeping posted about award deadlines, those kinds of things. Like CLN would get posted the work study, I think someday in August, and I would like mark it in my calendar, log on and see everything that day. So yeah, so one skill is just to like constantly be seeking, and then by statistically looking that much, you'll have some more opportunities come your way. Yeah.
Great. We're getting close to 1 o'clock. I wanted to summarize some very important advice around the balance of mental health, work, and school. So from Professor Bruce, you will always be able to see how many hours a week are required when you apply for a work-study position and what's the mandated upper limit. And so that connects beautifully to Alex's point. When you time management well and closely, you can figure out, is that a good match for what I have? And from Annetta, you can always meet one-on-one with a learning strategist to learn more about time management skills. And at New College, you would go to the registrar's office to get that kind of one-on-one support. And from what you've said, Alex, this is something you found on your own, but if you think that you could use some help, it's a skill. Time management is a skill. We do have folks to help you. We have one last question in 2 minutes, and this has to do, it's a great question, somebody else wanted to know this too, about our current climate. So we're living in COVID-19, we're living in a pandemic. Has it changed the approach for how many students might get to grad school? How would you say COVID-19 has affected your graduate school studies and your research? Have you been forced to slow down? And because you are in the program, what are you picking up? I know you aren't the profs in your program, but are you picking up that there's still going to be a vibrant grad student culture, or are things in more of a uncertain moment? Maybe we'll start with Jonathan.
So I think the culture between grad students will always be there. It's just that it will be moved online and not just in person. Like a lot of the conferences, the student council meetings are going to be online now. So I think the culture will still be there. But in terms of the amount of students entering into the field, it might be impacted if you are pursuing basic research, because in that case, you have to be in the lab in person doing the pipetting and everything else, which cannot be done, unfortunately, at home, right? Because you can't really bring home your lab and stuff. But if you are doing some like a coding or some clinical research that does not involve direct patient interaction, like maybe looking at some MRI scans or some of the EEG recordings that can be done offline at home, then it shouldn't be that impacted, in my opinion.
Thank you, Jonathan. Alex, what's your response to some of those ideas and issues?
Yeah, that's definitely what Jonathan just said is right. And, during this time, if maybe you're thinking like, I want research experience, try and look for, remote opportunities or skill development, like taking an online course in coding and those kinds of things, because statistical analysis and learning how to visualize data is a really important skill. So you can take advantage of that time. And I'm also doing basic research, so I still have to come into the lab. There's definitely like a backlog of experiments, but I think, yeah, the grad school culture is still gonna always be around, whether it's through Zoom or through being in the lab. I think it'll always be there. Yeah.
Well, both of you had the chance to kind of finish on a really optimistic note, right? We could have gone dark, but we didn't. It's been absolutely fantastic talking to you about your research, about your journey, about your experience. I enjoyed every single moment, and I'm sure the folks who participated did too. Yes, thank you, Colleen. Let's give our speakers all the applause that we can, because you were just fantastic. And I hope you enjoyed this month's New Research at Noon. Join us in December. We're going to have some more exciting projects to share. We will make sure that you hear all about them. And Alex and Jonathan, we wish you all the best for the great end of term. You did a great job, and I hope you had fun.
Thanks. Thanks for having us.
Have a great weekend, everyone. Take care.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye. Hey, Jeff, it's just you and me. How are you?